All right, on the recommendation of the last talk, I left my phone down in my chair in case my mom texts me, so I don't get stressed out. Oh, can you hear me back there now? There we go. All right, so this is just sort of a little exercise in identifying the distribution of sperm proteins in a genome that turned into a sort of serendipitous discovery of a neosex chromosome. So you probably know most of this already, that not all genes are universally beneficial to both sexes. Some genes provide fitness benefits to only one sex, and theory predicts that these are not going to be found randomly in the genome. Oftentimes they end up on sex chromosomes. And most of the support from this comes from XX, XY, sex determination systems. But of course, there's the big other sex determination system in animals, ZZ, ZW. And so I simply wanted to look at a couple of ZZ, ZW taxa to see if we see the same pattern there. Specifically, monarch butterflies and Manduca sexta, the Carolina Sphinx. So both of these Lepidoptera I chose because they have a really good data set that's minimal to this sort of work. They have sequenced genomes, they have good resequencing data, but most importantly, they have draft assemblies of sperm proteomes. So when I'm talking about a gene that has a sex-specific benefit, for the rest of this talk, we're going to use sperm proteins as a proxy here because this is a sex-limited cell type, so these are genes expressed in male-only cells. So just quick reminder as we go forward in the ZZ-ZW setup. Males are going to be our homogametic sex, so carrying 2 Z chromosomes, whereas females carry a Z and a W. So 2/3 of the Z's are found in males, so we can predict that sperm proteins, these male-specific things, are going to be more likely to be found on the Z than on the autosomes. The sort of proteomic part of this is beyond the scope of a 10-minute talk, but suffice to say that we got sperm samples from the males and got peptide sequences out of that via shotgun proteomics, and we're able to take those and map them back to the genomes to figure out what genes are producing these sperm proteins. And then the only real problem left is to figure out really what is Z-linked in our taxa here, because we have a lot of good data, but we don't actually have linkage maps for these lips. So I came at this from sort of 2 different angles. One that I'm going to call the coverage approach, and the other that I'm going to call the orthology count approach. So dealing first with the depth of coverage, the idea here is just that all else being equal, when you're sequencing something, the depth of coverage you get is going to be related to the amount of DNA you have in your sample. So across the autosomes, that should be equal between the sexes. But when we look at the Z chromosome, you have twice as much material there in the males as in the females. So we should see a 2-fold deeper coverage on the Z in the males compared to the females. And then the other method we used here is an orthology count, taking advantage of the fact that we've observed pretty high levels of synteny, which is to say sort of the ordering of genes along a chromosome, that tends to be conserved pretty well between taxa and Lepidoptera. So we can take a pair of species, one of our targets here, the monarch or the sphinx moth, and map it to count orthologs and predict orthologs between that and a species that we do have a linkage map for. In this case, Melatea, identified with the fritillary. So, starting with the coverage approach, first thing to notice is that this seems to work pretty well. It advances things too far. It gives us 2 discrete categories pretty much without any overlap. the one thing to note here with the axes on the bottom is this is a log 2 scale, so 0 down there is going to be an equal 1-to-1 male-female coverage. And that's where we see most of the scaffolds in our genome. About 90% fall in that equal coverage, which we're calling putatively autosomal scaffolds. And then the other 10%, we actually see up around 1, which is, again, in log 2 world, a 2-to-1 deeper coverage in males as opposed to females. So just using this approach, we can identify some putatively Z-linked scaffolds and test our prediction, as long as we can map the sperm proteins back to these scaffolds, which we can. Yep. So just looking at this, in Manduka, we see a confirmation of our prediction here pretty quickly. The Z chromosome is enriched for sperm proteins compared to the autosomes by a good little amount there. So that's all well and good. But what about monarchs? Well, something seems to have gone wrong here at first pass with our prediction, because the Z chromosome doesn't appear to be any better than your average autosome, or so it would seem. But like I said, I have this whole second approach for identifying Z linkage, and that was to use orthology counts. So the idea here is that we're taking scaffolds from monarchs which don't have a linkage map and trying to assign them to a chromosome in Melittea based on which chromosome has the most ortholog hits. And another good thing to check right off the bat is that it seems to work pretty well in general. You have larger dots represent more orthologs, and for each putative scaffold here, we have pretty much one big hit for most chromosomes, so there's a lot of sympathy between Danis and Melittea. But there are weird things like this. So here you have a scaffold that is putatively assigned to the 21st chromosome based on a large number of orthologs with the 21st chromosome in Melittea, that also, you can see on the very far corner there, has a good number of orthologs that fall on the first chromosome in Melittea. And the first chromosome, just sort of definitionally here, is the Z chromosome of Melittea. So it could be a couple of different things. That could be just some weirdo failure in assembly of our scaffold. Or it could be more interesting. It could be actually representative of some sort of fusion event between an autosome and the sex chromosome. So to try and tease this apart, actually pulled back from the depth of coverage approach, because that should still hold true. So if we walk down this scaffold, we should see a consistent 2-to-1 male bias in coverage, which we do, whether you're at the part that is orthologous to the Z or the part that has orthologs on Melittea's 21st chromosome. And so with that in mind, we can then split the Z into what I'll call the ancestral Z, that is things that map to the Z in both species, and then a neo-Z, which was autosomal in Melittea, but appears to become Z-linked in monarchs. And when we do it this way, we actually recover what we were hoping to see to start with, that the ancestral Z is enriched for sperm proteins, but the neo-Z is not. In fact, it would end up on the lower end of what's in the autosomal range for sperm proteins. And so basically what's happening here is if you take the Z as a whole, you're sort of averaging across these two and it's obscuring the enrichment that happened ancestrally because of this sort of dearth of sperm proteins on the neo-Z. So, as I was saying there, Manduka followed this pretty simply, and Danis Fletchbus did not at first, without taking into account this interesting event of putative chromosomal fusion, where an underrepresentation of sperm chromosomes fuse to what is an overrepresentation of sperm proteins, sort of obscuring this. So, wrapping up and sort of bearing the lead to what I think is one of the cooler stories in reproduction in Lepidoptera. The next thing and the next step for this would be to actually look across sperm dimorphism in Lepidoptera. So almost all Lepidoptera make both nucleated and anucleated sperm types. And it's something we've known for a while, but don't really have a good answer as to what the functional significance of these anucleated sperm are. So the next thing I want to do is actually try and repeat these results with this split between the dimorphic sperm, because all of the sperm proteins we identified here came from chromic samples of both nucleated and anucleated sperm. So, with thanks to the University of Kansas, NSF, and all of that, I'll take questions.
So you said your Neo-Z has a dearth of sperm proteins. Do you know what kinds of proteins are found on it?
Not as of yet. Have to dig a little bit into that more. Like I said, this is first pass. Like I said, we didn't even split the sperm proteins by type, the nucleated or anucleated sperm set. So.
Do you have any idea why the Neo-Z is depleted? Depleted in sperm proteins? Is it a coincidence, or is it actually something that's evolved? Like, can you look at the homologous chromosome in other species where sperm proteins are known, and is that also depleted?
Sure. I actually haven't done that yet. That'd be a good thing to do. I wouldn't a priori think anything super interesting is going on because, and I actually had a slide that didn't end up making it in here that's across the chromosomes. There is a decent variation in how many sperm proteins you find on a given autosome. So some have more, some have less. I wouldn't say that there's anything necessarily interesting happening there yet, but it is definitely worth looking into.
So the Neo-Z was shorter, right? And you said there was less of the sperm proteins on there. Could it just be that it was, since it was that much shorter, that there was less, and that if you scale it by length, that it was still the higher proportion relative to most of the autosomes?
This is by proportion.
Oh, sorry.
It might be a smaller target in that it is a shorter sequence, but that was all done by proportion and not just raw count.
Visual protein identification is how the things transcribed in sperm as haploids.
No, it's a pretty rough sperm protein from just whole cell shotgun proteomics, so we don't have that kind of sort of scale of resolution at this point.
All right, thank you.